Hi, and welcome to this lecture on environmental adaptation of animals. On these few videos, I will demonstrate how developmental biology has something to do with environmental adaptation. We have the same genes as we were having when we were born, but still we are not the same person. We have different size of the organs, we have different energy consumption in the organs, we have even different proteins in the organs as we were having when we were born. And we will start this video series with this phenotypic plasticity. Okay, here's the hooked seal, and all these seals, they are living underwater throughout the year, but they must give birth on a solid surface like on this ice cover. So newborn seal and the adult seal, they are living in different kind of environment, and partly it's because they have differences in the oxygen storages. So as you can see in this picture, there's much more oxygen stored in the adult, especially because it has so huge amount of myoglobin in the muscles. So it has about 2 times more oxygen that can be stored in the body, and this affects that. Okay, they can spend 90% of their life underwater, and well, these weanlings are remaining in the pack ice. And this diving is potentiated by these large oxygen storages. And actually, when comparing this, it's important to remember that, okay, it's not just because the adults are large and the newborns are small, but because it's over here, it's in milliliters per kilogram, so it's volume independent. But still, these weanlings are quite good in oxygen storage. So in humans, the average oxygen storage is a little bit less than 30 milliliters per kilogram. So when they are having limited oxygen storages, it affects on their behavior on the diving. Okay, when compared on the weanlings and adults, you can see that the weanlings are diving only a few minutes, and the adults are maybe 15 minutes. And the longest dives can be a little bit longer, but there is a huge difference between the weanlings and adults still. And also the depth of the dive is completely different. So the weanlings are living in the surface water, and these adults are living in the deep water. And then this limited oxygen capacity affects on the diving behavior, and then this affects on the hunting behavior. So they must get food on the surface. They can't have, they are not able to dive in the deep waters to hunt. And this makes this development physiology essential for their surviving. And as a matter of fact, the difference in this duration is not only that, okay, there was 2 times more per gram oxygen capacity, but actually these small animals use more oxygen. Smaller you are, more oxygen you are using per gram per hour. So that's why these weanlings are actually using these oxygen storages quite rapidly during this diving. Okay, so kids are not just small adults. The youngsters are different in the physiology, and also in humans. In humans, the brains reach the full adult size at the age of 7. But several other organs are drastically developing much later. And this affects that, okay, in the adults, maybe 20% of our energy is consumed in the brains. But in the kids, it can be 50%. And this affects then that, okay, if you are starving, when you are young. It will definitely have some mental effects. Okay. Development can even affect on the migration. Over here we have the stars of northern hemisphere. And so the stars are rotating, or the Earth is rotating, and then over here the stars that are not rotating are there on the North Pole. So most probably there's the northern star. And some birds are using these kind of images, like garden warbler, lehtokerttu. When they are migrating to south at night time, they must navigate by using the stars. And when they've been in the nest as newborns, they observe this rotation of stars and underground material chains to remember the stars in the center. And this helps them to navigate in the opposite direction. It's also affecting on the thermoregulation. So we know that, newborn people, humans, have brown fat, and that makes them easier to tolerate cold, but actually they can't tolerate it well. But then it's affecting especially on small animals with have very limited heating capacity. So this is a mouse in Northern America. The adults are able to maintain homothermy even in below zero temperatures. And the ability to form the heat increased during the first 3 weeks. So the newborns, they have about they can produce half of the temperature of the heat that is, whereas it is the animals that are already 3 weeks old. And at the same time, they change the insulation layer. So these newborns are practically naked. And then when they are getting the fur, the insulation is getting better. And that means that, now they can produce better heat, and then they can keep better the heat inside the animal. And this affects then that, now they can tolerate cold temperatures. So these newborns, naked mouse, they couldn't survive below 30 degrees of Celsius, but a week later they can survive 5 degrees of Celsius. And this affects that, now they can leave the nest. So when they are 3 weeks old, they can have time spent out of the nest because it's cold outside, and now they have the isolation, and they are able to produce the enough heat to keep them warm, etc. The enzymes also are different at different time of our development. This is the regular rat. Few days before the birth, these rats upregulate glycogen synthesis. So that's why when they are newborn, and they are able to store glycogen in, for example, in liver. And at the same time, when they are born, they start to produce enzyme that is needed for glucose synthesis. Now they can synthesize glucose and store the glucose as glycogen. And then, after the nestling period, they start to produce glucokinase that is needed to regulate the blood glucose concentration. And so enzymes are different, but also the tissues and what kind of cells they have. And so in some cases, when we were newborn or before that, we had the same proteins, but they were located in different areas, different cells, or even the cells were in different locations. And chloride cells are very typical in this example. And they are transporting chloride out from the fish. And in the larva of killifish, the chloride cells are located in yolk sac and on the surface of embryo. These cells are over here in green. And a little bit later, they are around the surface of the fish. And in the adult, they are only located in the gills. So you can have, before you have a decent gills, you are trying to pump chloride out from all the surface of the animal. Thank you.